Yeah, well, I'm Ronald Lane. I'm a neurobiologist. We have a technology that we're quite excited about for a variety of reasons, but we call the company HOPE-Neuron because we believe we have the capacity to deliver hope where today treatments don't provide that to individuals. And we're starting off with Lou Gehrig's disease, which is an egregious disease, a terrible disease that there's no treatment for it that's effective. And yet our mouse studies show that we are effective. And I have 10 years invested in this. So it isn't something that we came up with yesterday. But I'm a mechanic, and so I deal in mechanisms, and I'm not a clinician. We have clinicians involved, but that's not what I do. And my focus is if the mechanics are there, then I say, well, let's pursue it. If the mechanics aren't there, I say, let's not waste our time and money. And sometimes clinicians who don't really focus on mechanics will want to go forward with something they're excited about. But if the mechanics aren't there, I mean, you can do it, but in my opinion, you have a great risk of just investing time and money and getting nothing from it. So anyway, what I'm trying to say is that we're going with Lou Gehrig's disease, ALS, because we're finding a tremendous response in our transgenic mice that have the human ALS gene within them, and they die from it. All right. We buy these mice. They've been infected, transfected by laboratories that do this work. They sell the mice. We buy them, and the mice will die quickly unless they're treated. And we have a presentation that includes within it a bar that the mice get up on, and they run on it. It's sort of like a treadmill, but it's a tread bar, and they run on it. And if they don't get treated, they fall off, and then they die.
Oh,
Whereas if they get treated with our treatment, they run. They don't fall off. Now we have to treat them every couple of weeks. Then that translates, we believe, into doing it every 3 to 4 months in humans. And that statement is an assumption based on the science, not on our work. But we are set up to do our work with our technology at the Gregory W. Fulton ALS Clinic in Phoenix, which is part of the Barrow Institute. And it's number 3 in the world in its size behind Mass General and Hopkins for ALS. Anyway, the bottom line of that is that we're very, very excited about this potential treatment or this treatment that we think is potentially going to be very effective with ALS patients. Now, the reality is the mechanics that drive that is the cell talking and how the cells work with each other and break in one direction and enthuse in the other direction. Those mechanics are similar. The cause is not. The cause is mutational. All right, and every disease in this grouping has a tendency to have their own mutations. But the ALS mutations, once you get beyond the ALS mutations, you have the disease, the mechanics that continue to drive it forward that the patient succumbs to are very similar. They're autoimmune. They're very similar to the mechanics that are in MS, the mechanics that are in Parkinson's, and the mechanics that are in Alzheimer's. So frankly, our second goal is to leap over everything and go to Alzheimer's. And there's 2 reasons for that. It's by far the largest concern in America right now from the standpoint of expenditures. Americans spend more money on Alzheimer's than any other disease, all right? More than cancer, more than anything, all right, with Alzheimer's. And there's no hope.
Yeah.
the patients to succumb to that. And I would say that I could take that very personally because my wife passed with Alzheimer's. And so I lived with her, joyously living with her. And I say joyously because she was an incredible experience for me. 55 years I was married to that woman, and she was beautiful. And I went through that process. And I understand, therefore, the agony that
Mhm.
people suffer from when they get these diseases, in particular when they get Alzheimer's. And so our focus is to go from ALS and to demonstrate it. We're set up to do the human trial. We have a public offering to raise some capital, $5 million we're raising in a public offering, and it's called a Reg CF. It's a crowdfunding vehicle that allows anyone to invest. There's a dollar limit, and it's probably going to be very small, $1,000, maybe even $100. That the interested party could invest in, and we're limited. We're limited to 5 million, but that is fine because that 5 million allows us to do the human IRB, and that's the Institutional Review Board. It's reviewed by the institution that you're doing it in, by their board, and then they have to report the findings to the FDA. That allows us to very quickly do this ALS study at this clinic in Phoenix and get the results from that. And we believe we know what the results are going to be. What we don't know is what's the dosing that we have to use,
Mhm.
and then specifically how often do we have to treat. We can estimate it from the mouse work with formulas that the FDA approves, but we still need real data, and we don't have that real data. That is, how often do we have to treat? We believe it's going to work. The data says it will work, but the question is how often. So anyway, that's what we're doing. We're doing Lou Gehrig's disease. We have wonderful results in that. No one else has been able to accomplish this. And we're an anomaly because we're not a big drug company. We don't use drugs. All right. We use the cell's own immune system
Mhm.
that becomes dysregulated. And we assist the body to re-regulate its immune system. So the body then does what it's supposed to do. It kills the disease. We don't do that, or it manages the disease. We don't do that. It does, but we assist the body in doing that. And that's our technology. It's patented. We have more patents coming, more ideas on how to improve it and so forth and so on. But we're ready
Mhm.
right now to do the human trial for ALS with our technology and to start treating. And we think we can get all of this done. And actually, we believe, based on what the FDA is doing and has done recently, we could, with some a little luck, we could get this all approved and be commercially viable by the end of next year.
Yeah. Okay, so that sounds really impressive, and thank you for like the really detailed overview. Your story is definitely very touching. So I think for our viewers, you mentioned that you were like part of the mechanics side of it. So could you kind of explain how exactly the immune system is like dysregulated during ALS? And then also like what got you interested in ALS research?
Okay, let me answer your second question first, and then I'll get back to the first. All right. The
Hmm.
reason for ALS is not that that's what we were interested in the first place. Okay.
Okay.
The practical considerations with ALS. ALS is technically, with the FDA, considered to be a rare disease. And if it's a rare disease, they will give us accelerated approval if we demonstrate safety. That's all we have to do. We have to show that the patient is no worse off after we treat than they were before we treat. And
Hmm.
so we then can go forward and demonstrate that. Well, we are presently demonstrating safety in the mice. So we're very comfortable with what we're doing and why and the mechanics of what we're doing, why it's safe. But we've already demonstrated that in humans. We're set up to do a safety trial at Barrow, at the Gregory W. Fulton Clinic in Phoenix. We're set up to do that, the ALS clinic in Phoenix. Uh, And so the reason we went with that with ALS is that it is a disease that's terrible. The patients, frankly, die quickly in a relative sense, 20 to 40 months. They get dementia halfway through that. It affects not just the patient, but the family and the caregivers. It's expensive. It's just a terrible situation. And but technically, from where we're coming from, it is a wonderful opportunity to be able to demonstrate quickly for a small amount of money in a relative sense, a small amount of money to demonstrate that this technology works in autoimmune diseases. All right. So that's the first reason why we went with that. Now, the first part of your question was, how does it work? Okay. And how it is, when I started on this, well, let me stop there and say this, that the big pharma, the big companies, multi-billion dollar companies have spent $2 billion and oh shoot, a decade to try to solve this problem, and they haven't succeeded. And the reason is we're going to argue that they are using a method, which means drugs. They use drugs because that's what they do, right?
Yeah.
And I'm not arguing against drugs. I'm just saying that drugs will not work on this, and that's a whole nother story and a whole nother argument to go through. But drugs will not work here, and they haven't worked, as demonstrated by the tremendous effort and cost that the drug companies have put into solving this problem. Our solution was to say that we don't have $2 billion to spend like the drug companies. All right. And we don't have those resources of thousands, literally thousands of researchers to bring to bear to try to solve the problem. We don't have that. So my background is in comparative neuroanatomy as well as neurophysiology. All right. That's what my postdoc was in a laboratory at the University of Wisconsin back many years back. Okay.
Yeah.
Back in the 1970s. And it was a great lab. It was a great place. And they focused a lot on the comparativeness. What do I mean by that? The evolutionary, the simplest of mammals up to the very complicated mammals. And we worked on what things called tenrics. They were little, look like small porcupines, all right? They're just about this big, little bitty things, but they're mammals, all right? And so we worked on them. And when I say we worked on them, we worked on their brains, okay? It's all brain stuff. But we also worked with monkeys, and we worked with a lot of animals in between, all right? Comparative. We would compare one to the next, to the next, to the next, to the next, all right? So when I started looking at this, I said, all right, what is it that we're doing? And I'm pausing here to try to give you a quick answer, right? Because I got 20,000 PDFs sitting in my computers at my feet right here. Okay,
Yeah.
so I have to say that I'm compulsive is only a beginning. I mean, I really am after this, and I have the fortune that I don't have a boss. Okay, I can wherever my ideas and wherever my brain takes me, I can go there. All right, I don't need somebody's approval because guess what? It's my money also. I'm paying for it. So I don't need somebody else, and I don't need to beg for money and all that. Now, don't get me wrong. We all can use money, and we're raising money for that purpose. So to say that capital is I mean, this business is expensive. I mean, by any measure. But we are, I believe, we're set up to be able to do this in a relatively inexpensive method or fashion to show it and even get FDA approval quickly because we're a rare disease. All right, back to your question. How does this work? All right. The body is plastic in many respects. The immune system specifically is plastic. What do I mean by that? It's malleable, all right? And the cells, the immune cells are set up to do fundamentally, and it's a lot more complicated than this, but fundamentally they're set up to do one of two things. When it's time to kill, they bear arms and they kill. They kill viruses, they kill bacteria, they kill fungus, they kill. That's their job.
Yeah.
But when the killing is no longer necessary, they stop and they become healers and they heal. Same cell. Same cell will kill, same cell will heal. And the vernacular, the description that's used in the literature is to refer to the killing format, characterization is in M1 cells, standing for macrophage 1. All right. Macrophage is a white blood cell. All right. It's an immune cell. Macrophage 2, M2 cells, is the same cell that is now healing. It's not killing anymore. So when we have diseases like this, what happens is that because of the genetics, typically the genetics of the cells in the body of the individual, they get locked into the high propensity for killing type 1 cells.
Mm-hmm.
If you're locked into that, you get autoimmune diseases. And it gets real complicated. And I can go there, but this conversation would take a long time. but you've got these locked in M1 cells that are disease producing. So what we need to do is to signal the body to change and shift from M1 to M2 and to start healing. They've been destroying with M1. Let's get them over to M2s and so they heal. All right. Well, I want to make this simpler and quicker, and it's not simple. But this is what we do. There's a term called ephrocytosis.
Mm-hmm.
Cytosis is the action of a cell, of a cell. All right. Ephro means to the grave. All right. Well, why in the world would I raise that up? Well, because the mechanism that we're using is the mechanism that the two of you use, that Mark uses, and I use on a daily basis to the tune of 200 to 300 billion times we do this. Same thing, again and again and again. That's all part of normal physiology.
Uh-huh.
And what happens is when everything organic has got a lifetime. It's born, it functions, and then it dies. Everything organic, all right? So a cell is the same thing. It's born, it functions, and when its time is up, it dies. And in good health, the body then removes it. How does it remove it? Ephro, to the grave. It takes it to the grave, all right? And when it does that, there's a shift from M1 to M2. When it takes that cell to the grave, it is. Now, technically, what it does is it digests that cell, reuses it. Nothing gets wasted in nature. And in our body, that cell gets reused and comes back as an M2 cell. All right. Or it shifts, it shifts that macrophage, which is a type 1, will consume that apoptotic cell. That's what I'm talking about. It consumes it and it eats it. And when it does, it digests it. And then that becomes part of its meal for its next phase in its life in that immune cell. All right. Now, I need to back up and try to bottom line what I just said. Okay. Mm-hmm. M1 cells that get dysregulated that are a disease. We trigger those cells by presenting to them apoptotic cells, intentionally creating apoptosis, programmed cell death.
Yeah.
It is. We give cells that. They eat those. That's their job. They eat those. They get rid of those. When they do that, they then shift to a type 2 healing cell. And it's that apoptotic trigger that we're providing to the body massively. We put in 200 to 300 billion signals of apoptosis for the body to shift from type 1 to type 2. All right. Now, I probably have said way too much here and gotten way too technical, but I've tried to answer it. All right. There's type 1 that is a killing cell, but when it's chronic, it is disease. There's type 2 that's a healer, and you can't have both. If it's type 1 elevated, type 2 has to be down. Mm-hmm. So the only way you're going to get type 2 up is to bring down type 1. And that's homeostasis. That's the balance that we're after. And we create balance in the body using the body's own immune system. We don't do it. The body took 300 million years of evolution to create the system I was just describing. And we get out of the way and say, you cells know what to do. You just got a problem of imbalance. We're going to help you get rebalanced and get out of your way and let you make the body healthy again.
Oh, okay. Wait, I really appreciate that explanation. I don't think it was too technical, so it was really in depth.
I hope that is somewhat understandable because it is complicated, but I'm
Yeah.
trying to make it simple that it's a balance thing of the immune cells and it gets out of balance and our technology rebalances it so that it's normal health. That's what we do.
Hmm.
We don't use drugs. There's no chemicals that we're putting into the person's body. That's why it's safe. That's why we're very confident this is safe because we take the patient's blood, we talk to it. We call ourselves cell whisperers, right? We whisper to the cell, say, you need to go back there and heal it. So then we put it back in the body and they trigger the healing. They signal the healing.
Yeah, that's really interesting.
Could you maybe talk about some of like your major findings or breakthroughs throughout your research? Anything that particularly excites you?
Okay, you speak softly and I'm hard of hearing.
Oh, sorry.
Could
That's
you
all
talk
right.
about, yeah, could you talk about any like major findings or breakthroughs you've experienced throughout your research?
Well, I, let me say this. There's nothing that I'm doing that I'm using other people that I mentioned 20,000 PDFs at my feet. All right. That's what I use. I'm using other people's research that they've been doing for a long time. So I'm going back 100 years, frankly, of these. What I'm doing, and that in itself is not novel. Those are existing articles that are
Yes.
there that have been published by these researchers and they're in the public domain. What we're doing is that we're novelly, uniquely using those in a fashion that nobody else has done to construct this healing program.
Hmm.
That's what we're doing. And that's the novelty. And we have patents, and we're getting more patents on the processes that we're using. That goes way beyond anything I've described here. And it's complicated enough what I've already described, so I won't go any further. But that's what we're doing. We're using the knowns in a novel, unknown way. And that's the uniqueness of what we're doing. That's the invention.
So like drawing connections that people haven't seen before. I think this is like a really good pivot into your company. And so you mentioned that you're like kind of like triggering apoptosis or balancing these immune cells. So can you explain how exactly your devices will do that?
Yeah. Again, let me think for a moment and to give it to you in a simple fashion. Apoptosis is a product of a stressed cell. Okay, cells become apoptotic when they're stressed, and again, they respond to the signaling in a stress environment. Maybe that's a better way of putting it. When the cell's in a stress environment, they respond with apoptosis, and they then are eliminated. We already talked about efferocytosis. They're eliminated by the cell. Okay,
Yeah.
and so what we do is this is all sterile now. All right, we take and put in an IV port in the patient's arm.
Hmm.
It drains by gravity. We then drain some blood out, a half a cup of blood. Takes about 15 minutes for the blood to drain out, 10, 15 minutes. It goes into a flask. All right, just gravity feed into the flask. We then take that, disconnect it from the arm, all right? And when you disconnect it, it shuts off. We then take that flask, we take it over and we snap it into our device. All right. So we're a device-enabled therapeutic.
Oh, okay.
Okay, we snap it into the device. The device does two things fundamentally. It's called mechanotransduction. All right. What that means is that when the cells are exposed to a mechanical process, in this case, rotation, when we spin them, all right, and this is all computer controlled, we do it precisely. We know how many cells we're going to affect, how many cells we want, what we want with those cells, what we don't want with those cells, the kind of cells we want to produce, the kind of cells we don't want to produce. Because you can produce by doing this, you can overdo it and produce bad cells or harmful cells. Better way of putting it. All right. So we've got background and experience and all that, and it's all computer controlled. The operator doesn't do anything except hit a button. All right. It puts that flask in there, and the flask spins. Then we take and we dramatically increase the oxidative stress within that, and it's all computer controlled again. All right. And we know exactly the ratio of O2 oxygen and O3 oxygen. And we increase the number of oxygen atoms in that space while it's being spun. Okay.
Oh,
Those two things in combination will create apoptotic cells. And again, this is all computer controlled, so it's done precisely. If you do the same thing and it's not controlled, you end up with the croptotic cells that kill. We don't want that. We want cells that will heal. Okay.
mhm.
So A, you got to know what you're doing, and B, you have to control what you're doing, and that's our technology. All right. So we after we do that, the computer, we don't do it. They just put it there and step back, and the operator doesn't have to know anything about physiology. Just put it in there, hit the button, and step back. The computer does it when it's through. The light goes on. You take the flask out, elevate it, and by gravity, you reconnect it. The tube in here, by gravity, it flows back into the patient. That takes another 15 minutes. The whole process, the spinning of it, and that process takes about 5 minutes or less. That's the treatment. But getting the blood out is 10 to 15 minutes. Getting the blood back is 10 to 15 minutes. Now, when we're through, all of that apparatus that's involved, the tubing and the flask and so forth, that gets disposed of. It's used one time only. It's sterile.
Yeah.
So there's no prospect of it being contaminated. All right. It's all sterile. It's packaged. We use it. We set it up. We use it one time. We throw it away. All right. Or dispose of it because it's a biohazard technically. So we dispose of it. All right. So that's what we're doing. Now I hope I'm making some sense.
Yeah, that makes total sense.
Okay.
I think that's
Okay.
definitely really cool.
Yeah, so if like your research succeeds and if your device works, like what are some possible implications?
Like, is it gonna be
All
more
right.
accessible?
Yeah. Well, this is where it gets interesting. Mark and one of our wonderful coworkers is working on updating our presentation. When we get that done, hopefully sometime in the next week, we will send it to you. Okay.
Oh, okay.
It's a video presentation. It's got a narrative and it's got visuals and it shows the mice running on the tread bar and
That's
all.
exciting.
It's a wonderful thing. And it will explain, hopefully, visually, which is a real important method, and make some of the things I've been saying a lot more, we'll make them simpler, more understandable,
Mhm.
because you have the visual to look at it along with my verbal narrative, right? It'll be, I believe, a good piece for your edification, to explain to you what and why we're doing certain things that we're doing. And it's pretty comprehensive. I mean, we're not giving away trade secrets per
Uh-huh.
se, because at the end of the day, if the investor isn't protected, the investor won't help you solve the problem. So you need to protect the investor. What does that mean? It's called patents. All right. So there's a lot of law on what you can do and can't do relative to patents, and so we have very good lawyers, and we work within those boundaries. Okay, but we will send that to you, and that hopefully will make some things that I've been talking about more understandable.
Oh, okay. I look forward to that. That's really cool. I had like a question about I think you said like you use like an AIM platform. So could you explain like what that stands for or what it is?
Well, let me back up. Yeah, you also asked me, and then I will get to that. All right. You will also ask me about other applications,
Oh
right?
yeah.
I mentioned Lou Gehrig's disease, ALS, and why we're using it because it's a very rapid disease. It's a rare disease. We can get favorable treatment from the FDA. And I think that we have a shot, knock on wood, We have a shot of getting this approved by the FDA next year when we get the good results. All right. And we're going to invite the FDA in to be part of everything we're doing. They're going to be able to see what we're doing. But the history shows recently that the FDA will bend over backwards to try to help a group that's got a real technology with a shot at something that will work. They'll work over backwards with that group to help it work. All right. So that's where we're starting with ALS. I also mentioned that we're leaping over to Alzheimer's. And the reason for that, the time for that, is they are both autoimmune diseases. Different mutations, the etiologies are different, but they're both autoimmune diseases. Okay. So, and I won't get into the mechanics of it. I could do that. That's my world. I love it. But there's a strong argument that this will work in Alzheimer's. However, and the significance there is it's a terrible disease, number one. It creates a lot of fear in the family, number two. It creates dementia in the patient towards their end of their life, which is really a difficult situation, not just for the patient, but for the family, okay? It also, the mechanics of that also reach over to, and the presentation that we're going to give you, and that's really why, in part, I got off on that presentation. The presentation will speak to cancer, okay?
Wait, can you say that one more time?
Mechanics of this are also specifically, in fact, I got up at 4 this morning. That's what I do. And I'm sitting in my computer right underneath what I'm talking to you. I got articles on exactly what I'm going to talk about right now. Not my work, other people's work. But I get this and I say, that's true, and that's true, and that's true, and that's there. And then I confirm it with these other articles. That's true. Therefore, if those things are true, then boom, boom, and boom. And that means, where does it go? It goes to cancer.
Oh, okay.
It goes to breast cancer. All right. Now, tell me, if we have a treatment that we can demonstrate successfully that is remedial for breast cancer, is that important?
Of course.
Yes.
Huh? Is that important?
Of course.
Yes.
Okay, that's what we have. Fundamentally, there are two things that we're going to focus on: Alzheimer's and cancer.
Mm-hmm.
Now, Alzheimer's is a disease that one doesn't really, not concerned about. You're not worried about Alzheimer's, all right? Because you're young, all right? But the older you get over 60, you know people in the family, you know friends, you've heard of others that have Alzheimer's, and it's not a pretty disease. And the older one gets, becomes more of a real issue, okay? But it's age dependent. When you're young, it's not an issue. When you're older, it becomes more. And the older you get, the more it becomes. All right.
Mm-hmm.
I'm 81. I think of that. Now, I also know the disease, and I don't have Alzheimer's. I'm fairly confident I don't. All right.
Yeah.
I'm fairly confident I don't. All right. But it is age dependent. However, cancer, it's not. You know that cancer can strike an infant.
Yeah.
There is no age in our lives where we are immune to cancer. And so the market is huge. And if it's there and it's serious, and our focus initially is going to be breast cancer, because that is such a fearful thought, and it is not age dependent. Now, it tends to hit a little later in life, but it's not someone that is in their 80s necessarily. Can be, but it's not necessarily in their 80s. It can be a lot earlier than that. And of course, there are other cancers along the way besides breast cancer that this will also mechanistically be appropriate for, we believe.
Oh, okay. That's
Okay,
really
when I say we believe, I'm a scientist. We need the data. Okay. So we have two steps here. One is our regulatory CF funding, which brings in $5 million. It can't up to $5 million. We still have to raise it. All right. But we need investors to bring it in. And it's spent in two ways. One is it is spent today in the ALS trials to show that they work to get FDA approval. The second one is it sets up the filing for a regulatory A, not CF, but A filing, which allows us to raise $75 million. And that $75 million allows us to do the things I'm talking about here and specifically focus on cancer and those kind of things. All right. And so it's those two filings. We will also, though, in the first tranche of money, the 5 million, when we do that, we're going to expand our mouse work to include cancer. So we're going to have mouse models in that first $5 million that says, okay, we believe it's going to work in cancer. Boom, boom, and boom. Here are some models. And yes, it is working. It's doing the things it should do if it's going to impact cancer. And we want to demonstrate that. Now, we could spend a lot more than half of 5 million, which is 2.5 million. But that 2.5 million allows us to set the company up to take this forward. But in the first 2.5 million, we're going to do an expanded mouse trial that will include demonstration trials for cancer.
Oh, wow.
Now, let me say this, okay? The numbers on this become, I don't want to say silly because it's very serious things that we're talking about here, all right? But the numbers here, you know the size of the markets we're talking about, Alzheimer's and cancer. They're big markets. Yet we have strong data and strong arguments mechanistically that this will work on that. And therefore, the potential, I say the potential, for the investor in this. And by the way, I own a ton of stock. So, and I haven't had a salary in 10 years, okay? So, and I've been paying for the mouse work. So if there's anybody that's involved in it and committed to it and believes in it, it's me, all right?
Yeah.
But where I'm going is in this is I've got friends that say, okay, I'm interested. And I said, well, here are two things that I think that are a reason for you to continue your interest. I had a call early this morning from a wealthy man in Belgium, knowing what we're doing, and he wanted to know if he can come and meet. And I said, of course. So, it's there. I think there's a good, strong argument to say that the possibility of this being a wonderful return for an investor is there. And it's to the point of being almost unbelievable. So I don't talk about it. I don't talk about the numbers except to say, the size of the markets. If we're treating it, use your own intelligence to say this could be really economically, financially interesting, as well as beneficial to the patient and doing the good thing for the patient. All right.
Yeah.
Remember the name of our company, HOPE-Neuron. That's what we're doing. The most important thing we're doing is to generate and create hope for these people. But by the way, when we're doing that, like no one else has been able to do, when we're doing that, you can also make a little money or maybe a whole bunch of money. Okay.
Yeah, it's definitely really revolutionary, especially because your therapeutics are so wide reaching. And so I definitely believe is going to have a lot of potential.
Yeah, you are smart ladies. You're absolutely correct.
But I have like a little bit question. So do you have like any like intention to maybe make this treatment more affordable to patients or anything like any projects working towards that?
More affordable to patients.
Yeah.
Well, there's one drug right now, one drug approved by the FDA for Alzheimer's, ALS, right? One drug. It's applied once a year. All right. It's an IV treatment, right? Once a year they get an IV shot. Now it doesn't change the course of the disease. The patient still will move along and die. It doesn't change that. But it makes the patient feel better. It's paid for. It's approved by the FDA, and it's got a copay with it, but the rest of that is paid for by Medicare, by the government, by the taxpayer. All right. It's $174,000.
Oh, wow.
Okay.
for one shot.
That's a predicate to get to your question. All right. We believe that we can start because we have years of life and expenditures that we need to continue to pay for to move this along and make it a better process. So if we really squeeze it down in the charges, we won't have the money to make this thing even better. Because I think I can go way beyond the technology wise what I've been describing to you. I think I can go way beyond that in this application and in its comfort for the patient. All right. In other words, I don't think we'll have to necessarily be drawing blood from the patient in the future. I can do it differently. All right. But I need some capital to do that. And to do that, we need to take the capital that we get initially and apply it to improving the process. All right. So having said that, $174,000 for the one treatment that doesn't work other than it makes you feel better while you die. Okay. That's what's out there. The drug's called Endeavoron. All right.
Oh, okay.
And it's a Japanese drug. All right. It's a Japanese drug developed in Japan. They brought it to the United States. The FDA approved it without any U.S. clinical trials. They said, go. Okay.
Oh, aren't there
That's how interested and how committed the FDA is to addressing the ALS problem. Okay. Our model says, okay, we're not going to charge $174,000 for a drug that doesn't work. We're going to charge $55,000 for a drug that does work. How's that? Okay. Here's the issue. I've already said that the mice require continuing treatments. And I have estimated, don't have the data yet, estimated that based on the mouse work, that we will have to do that 3 to 4 times a year. If we do it 3 times a year, let me back up. We're going to charge $55,000, not $174,000, $55,000 for a treatment that'll work. Having said that, the good news is this. It will keep the patient alive, we believe, just like it does with the mice. Well, if that's true, they will need to get multiple treatments. And so the $55,000 is not the whole story. It's $55,000 each treatment. We think, we don't have the data yet. We think it will be 3 treatments a year. That's $165,000. But it's $165,000 for a drug that keeps them alive versus $174,000 for a drug that just makes them feel better while they die.
Yeah. So
Okay.
effectiveness is definitely more important.
Yeah. Yeah. And by the way, the $174,000, all but $40,000 is paid for by Medicare. There is a copay of $40,000 for Endeavoron. If you want that drug and you want to feel better, you have to pay $40,000 copay with it. Okay. I don't know. I mean, we're not approved. We haven't asked for approval yet. We're not ready for that. But I believe that based on the history of Medicare, we're going to reduce the cost for Medicare. All right. We're going to reduce the cost. It's a benefit to them. We can show the economics that with our treatment, the patient's better off because there isn't the buildup of all the other things that go with the outside treatment costs that go with ALS, with our disease, because the patients won't be necessarily confined to a wheelchair. All right. So anyway, I can go on and on about that, but I'm trying to answer the question that early on, we're going to be charging less for a drug that works versus the other drug that doesn't work. And we can give it away, but then we can't take this where we really want to, which means Alzheimer's and cancer. To do that, we need to continue to charge $55,000 a treatment, not $174,000, $55,000 a treatment. But we need to continue to do that.
Yes, okay, that's definitely really cool. And I'm so excited to see like the progress for your company. And so thank you so much for being here and just sharing your journey and your company. It's really, really inspiring for us to hear, especially because I've never heard about like these device-based like therapies before. So that was definitely very eye-opening.
Well, thank you for your interest and thank you for your time.
Okay, thank you very much.